DNA replication is a process in which DNA copies itself to produce two identical daughter DNA molecules. DNA replication is very important for the survival of species. And Michelson and Stoll proved by their experiment that DNA replicates according to semi-conservative model of DNA replication. For example, this is a DNA. When it undergoes replication, first both parental strands of DNA separates out like this, and then they will act as a template for the synthesis of new strand. So here new strand forms. So this is known as semi-conservative model of DNA replication, where half of the genetic material is conserved by each daughter molecule. DNA helicase is involved in separating the DNA strands by breaking down the base pairs of DNA so that two strands of DNA can be separated. DNA gyrase is also an enzyme which works slightly ahead the helicase and helps the helicase in unwinding of DNA and reduce the tension created during unwinding process. Single-stranded binding proteins which are attached to the single strand of DNA which prevent these strands to pair up again. So after this, these single strands will act as a template for the synthesis of daughter strands along themselves. DNA polymerase won't work unless some nucleotides are arranged at the template. So for this purpose, primase enzyme is involved to arrange RNA nucleotides at the template strand. And these short fragments of RNA nucleotides, which are 5 to 50 nucleotides, which may be variable within species, is known as primer. And it acts as a starting site for the activity of DNA polymerase. So primers are, you can say, initiators. And DNA polymerase start works after primase synthesize the primer. The Okazaki fragments are forming in the lagging strands which are interrupted by the primers. And this is the leading strand which is continuously growing toward the replication fork. And this is the discontinuously growing daughter strand in the form of short fragments which are Okazaki fragments and known as lagging strand. DNA polymerase 1 replaced the RNA primer with the DNA nucleotides DNA ligase enzyme. It formed the phosphodiester bonds. So this is how a DNA replicates. That's all about DNA replication. I hope you like this video. If you like this video, then do like and subscribe this channel.